Good afternoon, everyone. My name is Reynaldo Denard, and I'm presenting my project on analyzing the impacts of genetic and environmental factors on heart and lung sarcospan levels. During this research project, I was fortunate enough to work with my PI, Dr. Michelle Parvatiar, and on behalf of the Department of Health and Nutrition and Food Sciences. All right, so regarding this study, we know that passive and active cigarette smoking are major sources of cardiovascular and pulmonary dysfunction in the United States. And specifically, it can cause lung inflammation, emphysema, atherosclerotic dysfunction in the cardiovascular system. And so we are trying to focus on the negative effects of cigarette smoke. That's what CS stands for, on the myocardium, the heart muscle, and lungs. And we found that during smoking such said mice, we would find that the gene that we are focused on studying, sarcospan, which is SSPN right over here, we found that that would be downregulated within those mice. And so we hypothesized that pathogenic variants in sarcospan will increase vulnerability to chronic oxidative stress conditions. And so to examine this role, we took two groups of mice. We took knockout, which means that we knocked out the sarcospan gene all completely. And we took wild type, which just means regular mice that we would find in the wild. And we exposed them to 4 months of cigarette smoke. And then we would aim to find whether or not the role of sarcospan would maintain health. So just a little brief introduction. The cardiovascular system is vital for maintaining adequate and regional blood flow, and environmental factors such as exposure to cigarette smoke can cause decreased expression regarding not only the cardiovascular system, but the pulmonary system as well too. And the protein sarcospan that we are talking about today is found in the DGC, which is the dystrophin glycoprotein complex. All fancy terminology for it is a gene that encodes the structural integrity of the muscle cell membranes throughout the body. And so, again, we are aiming to see whether or not deleting this gene would increase or decrease cardiovascular and respiratory function. And so here's just a little diagram of sarcospan in the DGC. As you can see, sarcospan here is interacting with different proteins. We have dystrophin and sarcoglycans. And so to my methodology, we used a Scurex smoking machine. That is these two machines right here. And then we used 16 mice, eight wild type, eight knockout. And then of those eight, we would split in between male and female. And so the mice were placed into the Scurex machine right over here. And then we would place 7 cigarettes into each slot over here. And then we securely locked the machine. And then we would proceed to smoke them for about an hour and 30 minutes. And so we would do this 5 times weekly for 24 weeks. And after that period, we would utilize the echo machine at the College of Medicine. And then after we completed that, we would then sacrifice the mice to study their organs. And then that is where we have our results. So within our results, this is a picture of the wild type female mice. And so we have a picture of a room temperature, room temperature, I'm supposed to say room air, excuse me. Room air lung fibroblast and a smoked fibroblast. So this is the smoked lung fibroblast, and this is the room air fibroblast. And you can tell there's a drastic difference in its shape. In this diagram right here, you can see that the lung fibroblast is very thin, as you can hardly see it, but is very stretched out to provide support to nearby tissues. Whereas this photograph of a smoked lung fibroblast is very compact, and you can even see smoke residue on the lung fibroblast, preventing it from doing its function and spreading to nearby tissues, and therefore making room for pulmonary dysfunction. And then another test that we did, we used the Western blot technique to assess sarcospan expression in wild type mice. And so here, as you can see, we have a control group and we have our smoking group. As you can see in the control group, there is a greater level of heart sarcospan expression, but as opposed to the smoking group, you can see that it is very decreased, and even more drastically so with white adipose tissue that is just fat. And so in the control group for white adipose tissue, it is very prominent, but as a smoking group, it is almost non-existent. So that tells us that, okay, there is a role of sarcospan, and that exposure to cigarette smoke can in fact delete or reduce sarcospan expression. And so our last diagram here shows the assessment of cytokine response due to inflammation from isolated lung fibroblasts. And so as you can see, majority of the diagrams here, you can see that the knockout mice have a greater rise in cytokine response. And this is because this makes room for inflammation. And when you have greater when you have greater cytokine response, that makes more room for inflammation, which then makes more room for cystic fibrosis or other lung ailments, or in making room for more dysfunction and COPD, emphysema, things of that nature. And so we studied sarcospan deficient mice for several reasons. We wanted to see the variance in the sarcospan gene level and how it would lead to decreased expression, which is what we did in this situation. And also from prior research, we knew that reduced sarcospan expression would lead to an inflammatory phenotype, as we can see in this diagram over here. And it will also lead to greater susceptibility to cardiac injury. And also we wanted to investigate genetic factors in cardiometabolic diseases in the future, such as the link between genetic deficiency and metabolic alterations. So explicitly, we sarcospan expression, the disease Duchenne muscular dystrophy, and there's one that starts with the B. I can't remember the name, but those two famous diseases are known because of the lack of the expression for sarcospan. And so that can also lead to a future study in the future. And then also changes in fat tissue distribution and impact on adipocyte and heart function. And so within our conclusion, we saw that sarcospan deficiency did increase cardiovascular and pulmonary susceptibility to injury when exposed to cigarette smoke, which is likely due to impaired communication between the sarcospan gene and these pathways. And these are my references, and that is it.
Great. Thank you, Ronaldo. Do we have any questions from our audience? So Latika says, I'm always curious, what's next in your research journey?
So this study is also ongoing as well too. I'm still in the lab. We're still looking at other variables, such as what can happen when we introduce a high-fat diet. So that's what we're working on now.
Great. And Ronaldo, do you plan on going to graduate school, continuing research there? What are your future plans?
So after I graduate this semester, I'm going to go back home and take a gap year. And in that gap year, I plan on doing some research. I'm applying to specific positions like clinical trial coordinators and things of that nature. So hopefully I get to do some oncology research back home.
Sounds great. Yeah, there's another question. Going through this research, was there anything you learned that you were not expecting?
Well, I wasn't expecting to know how to dissect mice. It sounds kind of ridiculous, even though I'm doing this experiment working with mice, but I didn't think that prior to joining this lab, I would be dissecting mice. And It's actually pretty fun because I've showed my parents some photos, and my dad, he was like, "We need to call PETA on you guys based on how you guys are doing these mice." And things of that nature. It was pretty fun to get to work on mice, but yeah, I had no idea, well, not no idea, but I had no foresight into me working and dissecting mice, and that was pretty cool.
Awesome. And then there's another question from Chris. Can you share more about the smoking machine? It seems like a really interesting device to help you conduct this research.
So I'm still also learning more about this machine as we go on. This I'm fairly new to this lab, so I honestly can't give you an extensive breakdown, but essentially we would place the mice in these in this container over here. I don't know if you can see, but there's individual slots for each mice, and then we would have the 7 cigarettes in this slot over here, and then it would connect to this tube, which would go all the way and connect to this filter over here, and then through a set of processes would go into this tube over here and also introduce the smoke to the mice. And we would connect that to a computer, which would um, automatically go on to the next cigarette after it was finished, after taking like multiple puffs of each cigarette. So I'm sorry, I can't give you an extensive breakdown, but just an overall overview of the machine is what we did.
I think
You're
that
welcome.
might be it on questions. Thank you, Ronaldo, for sharing your research with us.
Thank you, and thank you everyone for listening in, and thank you for having me.
Great. So our next presenter is Parker North. Parker, whenever you're ready, you can share your screen with us.
Alrighty, so good afternoon. My name is Parker North. I'm a co-manager of the mental health team within the Global Health Collaboration Project here at FSU. And on behalf of our team, I'm presenting our study on the prevalence of adverse childhood experiences and the relationship to violence and substance abuse among university students in Honduras. And so adverse childhood experiences, or ACEs, are potentially traumatic experiences in childhood and adolescence. They include but are not limited to physical abuse, sexual abuse, emotional abuse, neglect, parental incarceration, parental substance abuse, parental divorce, housing instability, and food instability. And adverse childhood experiences are unfortunately not an insignificant problem. It's estimated that globally they have a $14.1 trillion annual economic burden. And ACEs increase the risk of developing chronic diseases such as cancer, stroke, type 2 diabetes, obesity, kidney disease, and respiratory disease. Adverse childhood experiences also increase the risk of engaging in health harming behaviors such as risky sexual behavior, smoking, teenage pregnancy, and violence. And since ACEs inhibit development in childhood and adolescence, victims are less likely to have the ability to cope with stress and therefore more likely to engage in violence. And so there's a generational cycle of adverse childhood experiences and violent behavior. And so research shows that adverse childhood experiences are more prevalent in low and middle income countries like Honduras, which is where the study is. Additionally, Honduras has a severe history of violence due to drug trafficking and gang activity. However, there's only been one study on the prevalence of adverse childhood experiences in Honduras. Therefore, research into the risk factors for ACEs is critical because without an understanding of these risk factors, we can't develop effective prevention strategies. And so the first objective of our study is to assess the prevalence of ACEs, substance abuse, and violence in our sample. And then we're going to see if there's a relationship between each of these variables. And based on the global literature, we expect a positive correlation between ACEs and violence and between ACEs and substance abuse. The second goal we have is to add to the ACEs literature on low and middle income countries. These countries have little data on ACEs, despite experiencing some of the highest risk factors for ACEs. And so our study will provide crucial literature in this area. And a tertiary objective is to provide data to guide the creation of evidence-based ACEs prevention strategies in Honduras. And so for our study design, it was approved by the FSU College of Medicine's IRB and the Universidad Nacional Autónoma de Honduras, or UNAS, their IRB as well. and the study was administered at UNA in Tegucigalpa, Honduras. And then all questionnaires for the study were translated into Spanish by medical professionals in a culturally competent manner. The Adverse Childhood Experiences questionnaire contains 10 yes or no questions about 10 different adverse childhood experiences, and it also asks the impact of ACEs on the participant as a whole. The Maudsley Violence Questionnaire, or MVQ, is 54 true or false questions assessing macho attitudes and acceptance of violent behavior. And so machismo or macho is an idea of masculinity prevalent in Latin America. In general, it accepts men's domination and violence towards women and encourages male emotional suppression. And so from the MVQ, there are 3 scores: a macho score, which measures the participant's view of machismo, an acceptance score, which measures the participant's acceptance of violence, and then a total combined score. And then the last questionnaire we administered was the Tobacco, Alcohol, Prescription Medications, and Other Substances, or TAPS questionnaire, and that measures the frequency of use of tobacco, alcohol, illicit drugs, non-medical use of prescription drugs, and other substances in the past 12 months and 3 months using that yes or no questions. For our sample, to meet the inclusion criteria, individuals must be attending UNA, be Honduran citizens, be 18 years of age or older at the time of the study, complete the informed consent process, and be able to complete the questionnaires. And so our sample consisted of 287 randomly selected students who met the inclusion criteria. And for our data analysis, we first calculated descriptive statistics and then used Pearson R correlational analyses for the relationship between the ACE score and the MVQ scores, and used Spearman correlations to test the relationship between ACEs and TAP scores and between MVQ and TAP scores. And so now on to our results. So 86% of participants reported experiencing at least one adverse childhood experience, and 60% experienced at least 3 ACEs. And according to the CDC, adults with an ACE score of 4 or more are 1,220% more likely to attempt suicide, 1,000% more likely to use injected drugs, 460% more likely to have recent depression, and 390% more likely to have lung disease. So there's a significant proportion of our sample who are at risk for all these health outcomes. And the bar graph on the right shows the distribution of the number of ACEs reported by each participant. And so the highest number of participants reported 3 ACEs. This next graph shows the prevalence of each of the ACEs measured. And so the most common ACE was verbal abuse at 60%, and the second most common ACE was physical abuse at 45%. And from our statistical analysis, it showed that ACEs and MVQ total scores were significantly positively correlated, as shown by this graph. And from the TAPS questionnaire, substance abuse was mostly absent, and when participants did use substances, participants reported using them less than monthly. However, there were significant findings between substance use and ACE and MVQ scores. Specifically, any tobacco use was positively correlated with MVQ macho and MVQ total scores. Any drug use was positively correlated with the ACEs total score and all 3 of the MVQ scores, to be macho, acceptance, and total. And any non-prescription medication use was positively correlated with ACEs total score and all 3 of the MVQ scores. And so the literature shows a high prevalence of ACEs in low and middle income countries compared to high income countries. And our findings agree with this. 86% of our participants reported 1 ACE, which is higher than the global average of 60%. Furthermore, 50% of our respondents reported that their ACEs negatively impacted their health, which agrees with the literature showing the negative health effects of adverse childhood experiences. We also found a positive correlation between ACEs and violent behavior, and this may help to explain the high rates of violence in Honduras. From our results of machismo, we found that men have higher machismo acceptance scores than women. We also found that ACEs are positively correlated with higher machismo scores, and that ACEs and machismo scores correlate with violence acceptance. And our findings on machismo agree with the other literature. Additionally, our findings agree with previous research that those with more adverse childhood experiences are more likely to engage in substance abuse. And some of the strengths of our study is we're the second ever study of adverse childhood experiences in Honduras, the first with a young sample. We're also the first to analyze how ACEs connect to violence and substance abuse in Honduras. And we also underwent a rigorous ethical review process as we had IRB approval from 2 universities. Some of the limitations, we didn't have any qualitative factors in our questionnaires. They were all multiple choice or yes or no questions. There are limitations with the ACE questionnaire itself. Each ACE on the questionnaire has an equal value, and also participants cannot rate the significance of each ACE in their life. They can only fill out a question at the end that assesses the overall significance or impact of ACEs on their life. Another limitation is the study population is of educated and urban participants. Research shows that educated persons and people in urban areas are less likely to experience ACEs. Therefore, our study may underestimate the prevalence and severity of average childhood experiences throughout all of Honduras. And then the last limitation is we didn't examine any correlation between ACEs and resilience. And so we cannot make any conclusions about how ACEs may affect long-term behavior. Summarize the results, 86% of Hondurans in our sample reported at least one average childhood experience. And this is an important finding because it's much higher than the rate in developed countries. For example, the rate in the United States is about 60%. Higher ACE scores were positively correlated with higher MVQ scores and drug use. Therefore, ACEs prevention strategies could lower violence in Honduras and improve quality of life. Future research should address this limited sample of our study. Again, our study only focused on urban educated Hondurans. Therefore, future research should study rural and uneducated Hondurans, which is the majority of the country. Future studies should use questionnaires and interviews or short responses that have qualitative measures. That way we can gain a more comprehensive understanding of the risk factors for ACEs. Additionally, further research could examine resilience and other behavioral factors in order to draw conclusions about the long-term behavioral effects of ACEs. And ultimately, by using population-specific measures, we can have a better understanding of how different Hondurans experience ACEs, thus paving the way for effective ACEs interventions. And here are my acknowledgements. Thank you to Dr. Charles Fleischer, my faculty mentor, to all of the other investigators on our team. We're an interdisciplinary team between FSU, FAMU, and UNA. And special thanks to the FSU College of Medicine Center on Global Health.
Awesome. Thank you, Parker. That was really interesting. We can open the floor now to some questions. Okay, so our first question, in doing this research, was there any concern of survey bias? For example, if a survey respondent did not recognize that they were a victim of ACE? If so, do you think this would be addressed with a qualitative component in the future?
Yes, so that would be a concern. It's be a concern broadly of ACEs research because there's a pretty standardized questionnaire from the CDC that's been used since the Kaiser Permanente study in 1995 that kind of published the idea of average childhood experiences. And that is one downside of that survey would be survey bias. And you're exactly right. Some qualitative components with the interview or mixed methods would definitely help to rectify that survey bias. And there's also bias in interviews. So I do believe it is kind of a give and take.
Great. Next question. This may be out of your scope on the project, but I'm curious about the ACE prevention strategies. Do you know what any of these strategies are?
Yes, that's a great question. So FSU's actually center on child health. I believe that's the name of it. I might be messing up a little bit. Has great resources on this if you're interested. They do work. I actually got to talk to one of the researchers in Immokalee, Florida that works at FSU, researching average childhood experiences and toxic stress. So that's the toxic stress aspect of ACEs, which we didn't really study here. That's what you target in the prevention strategies. So that's basically the idea that when you experience these ACEs, right, like abuse, neglect, parental incarceration, violence, etc., your stress response system is activated, right? Which is when it's chronically activated, that has negative health effects, right? The common expression people use to describe it is if you're walking through the woods and you see a bear, right, you're going to be very excited, right? Your body's be full of adrenaline. That's good for you. That way you can run away. But every time you go home, there's a bear, and that, right, that's going to have a negative health effect, especially for children, right? Because their brains and nervous system are still developing. So most of the prevention strategies are going to target that, trying to cope with how to properly respond to stress. Include CBT is most common. Cognitive behavioral therapy is the most evidence-based way to process PTSD or different forms of trauma and chronic stress. And so most of those prevention strategies, in short, are going to use CBT.
Great. Next question. This seems like an interesting but potentially pretty traumatic subject matter to hear. Was there any sort of preparation for you as the researcher?
That's a good question. I'd say for this project, not specifically, only because I've researched ACEs in the past. I'm part of the IMS program. That's my major at FSU. And for my capstone project, I'm doing a secondary data analysis of ACEs. And how they relate to socioeconomic status. So I've had learned about ACEs and been able to have experience researching them before this project. So nothing particular with this project about preparation, but that is an important thing, especially when we're bringing on new people to the project, is to help give them a full understanding of what we're going to be engaging in. That way it can have, just like we have informed consent for participants, we want to have informed consent for our researchers. That's a great point.
Great. Another question here. What made you want to get involved in this particular research project?
Yeah, so I've researched ACEs in the past. I got to go down to Immokalee after my freshman year. I just finished my junior year now, so 2 years ago, where I had to talk for a couple of hours actually with one of the researchers there who was researching ACEs, and that's how I learned about them. And just been really interested in the topic ever since. And so I jumped on board with my capstone project last year, and then I joined this project back in January. And it's been really exciting. Obviously, it's we don't want to have to research this topic, but given that we are, it's interesting to me, and I'm really grateful that our research can actually directly benefit communities.
Great. And then Latika says, cool project. Did you get to travel to Honduras, or were you able to conduct this all remotely? Also, do we anticipate there was any or much under self-reporting, as these were very sensitive, possibly incriminating topics?
So yeah, for the first question, so our project is again multi-institutional between UNA, FSU, and FAMU. So we have part of our team is in our medical students in UNA or our medical providers there, and so they were the ones that actually conducted the study or administered it last fall. But normally the students from FSU and FAMU do take a trip every year. Unfortunately, that got canceled this year, but we're hoping to go back at minimum next spring, if not also this fall as well. So normally we do, and like when we go back next spring, we're already working on IRB approval for a follow-up study. We're trying to figure that out. We're in the early stages there to administer the next study, but we didn't administer this one in Honduras.
Great. And then lastly, what are your future plans? Do you plan on continuing with this research?
Yeah, so I'm now the co-manager of the team. I joined back in January, and I was chosen to be the co-manager of our team 2 months ago. And so for this research, I'll be presenting it at a poster session at the National Conference of the American Academy of Family Practice, at their national conference in September in Phoenix. We're writing the manuscript for this paper right now, and then so I'll be continuing with this research at minimum through May of next year. I'm applying to medical school right now. I graduate in December, so I'll be starting med school either in May or August of next year, hopefully.
Awesome. Oh, we have another question here. Would you think of expanding this research to any other countries?
That would be great to do. Our project is fairly focused on Honduras since we have most of our infrastructure is there, our connections. And so we're pretty committed to Honduras, but there's great researchers who are doing ACEs research throughout the world. It definitely needs to be done in low and middle income countries where there's that big disparity.
Great. Last call for questions for Parker. Give it a few more seconds here. No questions. Okay, thank you so much, Parker. We learned a lot. Thank you for sharing your research with us.
Thank you.
Our next presenter will be Miss Tracy. Tracy, whenever you are ready, feel free to share your screen with us.
All right, can you guys see me all right?
Yes.
All right, perfect. So let me start. All right. Hi, my name is Tracy Muhammad Usher. I'm a senior majoring in biology, and this project is co-sponsored by Dr. Pearl Rivers-Key and Dr. Emily Duval. Today, I'll be talking about a phylogenetic analysis of plasmodial prevalence in wild birds. So malaria is a mosquito-borne illness that's a result of hemoparasites of the genus Plasmodium. This disease is particularly devastating to human populations, killing about 2.7 million people annually. The vast majority of these deaths are located in the African continent, and 9 out of 10 of them occur in children under the age of 5. Symptoms of malaria include anemia, lethargy, fever, and in extreme cases, seizures, and even deaths. And while this number is slightly lower because of antimalarials, this is still a humanitarian crisis. However, humans are not the only animals to experience malaria. Bats get malaria, as do lizards, so do antelopes and other bovines, and finally, so do wild birds. So what's avian malaria? It's almost exactly like I was talking about with humans, in that they are hemoparasites of the genus Plasmodium. This is a gram-stained slide of avian blood, and the nucleated blood cells here are stained purple. The hemoparasites that are attacking the blood cells are stained light pink. Just like in humans, it's a mosquito-borne illness. And the 2 most common vectors are found in the Culex and Aedes genus of mosquitoes. And the symptoms are pretty similar. However, they're slightly variable across all avian taxa. So while some experience just lethargy and fatigue, others just skip completely over that and just have extreme cases that result in death. So my focus is on avian malaria because this disease poses substantial threat to island archipelagos, specifically so in Hawaii, but now also in the Galapagos and New Zealand. In addition, other things like pathogenicity, fitness, and such are relatively unknown. And given that this is becoming such a huge conservation issue on island archipelagos, more research needs to be done as to the factors that contribute to disease prevalence. And to emphasize that point, I want to share with you that of the 10 avian species that were declared extinct by the US Fish and Wildlife Services in 2023, 8 of which were Hawaiian endemic passerines, which means songbirds that only exist in Hawaii, exist nowhere else, and are now declared extinct. And while there are likely other factors that contributed to their demise, habitat restriction of available habitat as a result of malarial prevalence is likely a large or a huge contributing reason as to why these birds no longer exist. And so our research question was, how does evolutionary relatedness predict plasmodial prevalence? And so what is evolutionary relatedness? I might sometimes say phylogenetic signal as well, so I'm sorry if I switch that up. This is a slightly inaccurate clade or phylogeny of the clade vertebrata. And so this is a representation of vertebrate species here. And I'm using this to show you what a phylogenetic tree looks like. Essentially, this depicts like a brief tree of life almost, and it depicts the common ancestors between all of the tree of life. So where these branches branch off represent different groups of species, and where they connect show a most recent common ancestor. And so I'm looking if certain portions of this clade are more or less likely to have plasmodial prevalence, i.e. avian malaria. And so now that we've defined what evolutionary relatedness is, we're going to define prevalence. Prevalence for us was defined as the number of infected individuals over the total number of individuals measured. So to be really clear on this topic, here's a little subset of Neo-Aves phylogeny. And I'm basically trying to look at if certain groups of birds are more or less likely to contract avian malaria. So our methods, we first define prevalence, and then we reference journals from a systematic meta-analysis of plasmodial infection. After I combed through the data that these researchers were kind enough to send me, I looked within each journal and parsed through individual species. So I basically took each species name, how many species were measured, and whether or not these species did or did not have plasmodial infection. From there, we referenced the literature to generate a tree of the species that we were including in this study, and then using R, we ran a model. So we have 3 different measures of evolutionary relatedness / phylogenetic signal. The important thing to note on the first 2, Pagel's lambda and Blomberg's K, both of these are used commonly to, of course, measure evolutionary relatedness, but the mathematical difference between the 2 is different enough that both are commonly run in these types of studies, and then their values are slightly different. We also included a linear mixed model to include characteristics from the data set on the top left called AVANET. We were trying to investigate whether things like migration, primary lifestyle, and trophic level were likely to correlate with plasmodial prevalences. And those characteristics were included in the original meta-analysis we were referencing. In addition, we also excluded all data points where individuals were less than or equal to 10. So we'll start with some summary statistics. I know I just showed you a phylogeny that was linear, and so this might look a little bit confusing, but I hope that you'll forgive me because I think it fits the screen a bit better. This is a circular phylogeny of the 91 species we included in this study. And so you can see that there are small little groups created within those, some of which by family or whatever, and we're essentially looking at whether or not those groups are more or less likely to get avian malaria than is predicted by chance. So as for our results, we first looked at Pagel's lambda. Our p-value for this was 0.284. And if you know anything about statistics, we adopted a standard alpha of 0.05 here, which means that we rejected that alternative hypothesis. In addition, this is Blomberg's K, and so our p-value was 2.75. So again, that was rejected for insignificance. And then finally, our generalized linear mixed model, which took into account migration, trophic level, primary lifestyle, in addition to things like inconsistent sampling efforts, were also insignificant. And so our take-home values here were that phylogenetic relatedness did not predict plasmodial prevalence, which to us was really confusing because in many range-restricted studies and in certain montane regions, other researchers did find that phylogenetic relatedness did predict avian malarial prevalence. And we think that this could be because the sampling efforts are just too limited right now to properly measure on a global scale whether a phylogeny could predict this. And then finally, our next steps for this project are to include Hemoproteus prevalence. So Hemoproteus is an alveolate, just like Plasmodium is, and produces a very similar malarial-like illness. Within that, we are trying to see whether that more specific parasite experiences like slightly more coevolution with their host species, and as a result, whether or not phylogeny could predict its prevalence. And that's where the project is going. Big thank yous to the Duval Lab. This project originally started in like a spring DIS season last semester, and so without her help, I couldn't have gotten this started. The researchers who sent me their meta-analysis, but specifically Dr. Weilin Yan, was instrumental to getting this up and running. Thank you to the CRE for letting me present this. I referenced all these images from Wiki Creative Commons. And then finally, Dr. Pearl Rivers Key has been monumental in getting this going, not only for emotional support, but also for her sage wisdom in getting this analysis up and running. So thank you.
Thank you, Tracy. Congratulations. And we are opening up the floor for questions now. Any questions? Okay, we have one here. What made you want to get involved in this research?
Um, so beyond the conservation concern, which I hope I outlined adequately enough, I'm a Native Hawaiian, and so the thought of a lot of these endemic species going extinct, and then more specifically, the fact that I'm studying to be an ornithologist, the idea of not doing a project related to a huge conservation concern that the islands are experiencing would be kind of shameful, I think.
So Latika, fascinating research. Just confirming, I still do not have to worry about getting malaria from lizards or birds. Still just mosquitoes, right?
As of right now, I would say I'm no expert on human malaria. But the plasmodium that infects us is of a different species and infects lizards and birds and such. And it doesn't typically make those types of species jumps.
Great. And then Dave says, I know you have also conducted a good deal of field research. How was it focusing? How was it focusing on mainly statistics with this study?
It was a fun change of pace. It definitely made me hone some R skills a little bit more than anything that I've done this far. And so now I'm a much more confident programmer for sure.
Great. Any other questions? Okay. Warren says, knowing that you want to continue in ornithology, are you planning on continuing this research subject or some other subject?
This topic I think is really interesting because of those studies that I mentioned before, where in or like geographically restricted regions, they do find that phylogenetic signal is correlated with avian malarial prevalence. And so the fact that we didn't find that is puzzling, and that is really exciting, I think. So I do want to continue to expand this data set. And while a meta-analysis doesn't exist for Hemiproteus prevalence, I'm hoping to do that myself and compare the two parasites to see if there is signal between them. In future, I do think these analyses could be rerun if we had more information on plasmodial prevalence. And I do think hopefully that with more sampling effort, our results might look a little bit different. As for continuing this in graduate studies, I don't know. I like animal behavior. So this was kind of maybe just a one-off because it's not super related to like the field work David mentioned that I like doing.
Awesome. Thank you, Tracy. Any other questions from our audience before we let Tracy go? We will move on to our next presenter. Thank you so much, Tracy.
Thank you.
And our next presenter is Catalina. So Catalina, whenever you're ready, you can go ahead and share your screen with us.
All right. Hello, my name is Catalina Casillas. I am a third year at the Florida State University. I'm currently majoring in international affairs and French. And this past academic year, I got to participate in the undergraduate research opportunity program. And I conducted a research project under the supervision of Mr. Damien Bolt. And this project was actually part of a larger project that he was working on, but my specific role in the project was investigating strategies of denial in response to targeted mass killings committed by governments across the world. It is a pretty morbid topic from the title, but I think it's honestly pretty cool and also pretty enlightening. So let's get into it. And so I wanted to give a little bit of background on why we're researching this topic. There is a lack of data currently that prevents researchers from systematically examining strategies of denial in the context of human rights abuse allegations. And what this study aimed to achieve is collecting quantitative data on how political leaders deny human rights allegations in events of targeted mass killings, which from now on will be referred to as TKMs. And basically, we're doing this project to understand how denial is used to better counter anger and shame when human rights advocates shame the state, which basically triggers anger and negative emotions in target states, which can in turn cause a backlash counterproductive to rights efforts. Next, I wanted to define some key terms that we use in this project. And so denial is a communication strategy used by political actors, which are public declarations of human rights violations that are untrue. Moving on, a shamer is an actor that accuses a country of committing human rights violations, and this country in question is called the target or the target country. And then we have two main strategies of denial. We have offensive denial, which deflects accusations back onto the shamers, and then defensive denial, which basically maintains that any accusations of violence are untrue or that violence is legitimate or necessary. And so moving on, for the methods of our study, or my study, it was pretty simple. So our research mentor, Mr. Damien Bolt, gave us a list of case studies bi-weekly, and for each case study, we would follow the same method. So for each study, we would start out with a basic 15-minute search just to gather a little bit of background material on what the case study might be. And then once we were familiar with the actors involved in the case, we would go on to Nexus Uni, which is a library database provided through FSU, and we would scour articles through the Associated Press or New York Times for evidence of denial. And if this preliminary search did not yield any promising results, we would also go to outside sources. Secondly, after gathering our sources, we would evaluate whether political leaders used a strategy of denial. And then we would record this onto a master Excel sheet that lists each variable, and we would code each case according to a code book that I'll go into greater detail in later. And then to ensure accountability, we would record our sources by downloading them and then making note of the key search terms that we used. And so this is a pretty hefty slide. I'm not going to go through every variable that we used within our code book, but the main ones would be if there's evidence of denial, then we have our two main strategies, which are offensive and defensive denial. We would also note if political leaders would shift responsibility of the TKM onto other actors, whether racial rhetoric was used, whether terrorism or sovereignty were called into play during this denial. And so we would account for a pretty wide scope of variables that political leaders would use in their denials. And so in order to better demonstrate our methodology, I wanted to give an exemplar case. And so I'll be going into further detail on the genocide in Darfur, which is a region in Sudan that took place from 2003 to 2007. And so, like I said, Darfur is a region in Sudan, and it was essentially the military-led government and the proxy militia called the Janjaweed pitted against non-Arab rebel groups. And during this event, over 200,000 civilians died, and another 2 million were displaced from this region. And so in our master Excel code sheet, we would account for the name, the location, and then of course the start and the end date. And in my research on Nexus Uni, I found an Associated Press International article that basically gave us clear evidence that there was denial. And so the foreign minister of Sudan said that there's no genocide in Darfur. And then the president of Darfur also gave us evidence of defensive denial when he said that reports about the genocide were fabricated and carry no truth. And he also ended up shifting responsibility of the violence onto tribal conflicts. And so in our coding sheet, we were able to code for evidence of denial. We were able to code for defensive denial, a blanket and that a blanket denial of any violence that had occurred, and then also shifting responsibility. We would also account for the method, which would be later public statements by the president and by the foreign minister. Moving on, we found other Associated Press articles that included racial rhetoric targeted towards Jewish people, and also made a claim to sovereignty, stating with the America's department head stating that this matter is not the business of anyone else. And so we were able to code for racial rhetoric and also claim to sovereignty. And then we would also record the titles of the files that we saved of the sources just to ensure replicability. And so the conclusion of this project was that we found that denial is a very common response to human rights abuse allegations, and it is used in more than 80% of case studies. In terms of variables like strategies of offensive denial, defensive denial, blanket denial, racial rhetoric, and appeals to sovereignty alter instances of denial case by case. Defensive denial was the most common form of denial at 60%. I also wanted to go into the implications of this project. And so while the overarching project that Mr. Bolt is doing is still ongoing, this research contributes to a systematic analysis of public opinion on denial, as well as strategies of denial in an ever-expanding field of human rights. And so just some acknowledgements. I would like to thank Mr. Damien Bolt for his supervision and his mentorship and his guidance. And I also want to thank my Year Up leader, Natalie Escobar. And then I did have a coworker, Devin George. And so I also wanted to thank her for her contributions to this project. Thank you.
Our first question is, was this study limited to any particular geographic region or time period?
So we had a pretty wide range of cases. We weren't limited by geographic region or time period. I think we were limited by time period in the fact that we needed to pull articles from reputable news sources. And so I think our earliest case probably started, I would say, the earliest case that I remember researching was maybe in the 50s, and that was still hard to find sources on. And then in terms of total cases studied, I think me and my partner were able to code for about 60 cases in total. And this project, like I said, is still ongoing. And so as this project is passed on to other Europe students, I'm sure that this number would grow.
Yeah, so you talked about total studies examined. Also, were you mostly looking at past events that had enough time since occurring so that the detail or facts were clearer or more consensus, or had more consensus, or was this approach also used or useful for current events?
So we didn't focus on more current events. I think the most recent events would stretch into maybe the 2010s. I think that this approach was honestly like the database that we had been told to use was more useful for more recent events. And so it was easier to come to conclusions on more recent cases. However, like I said in my earlier discussion of methods, if our search through Nexus Uni didn't yield many useful results, we would go outside of the database to sort of find outside information.
Great. And what's next for you in your research journey?
So I worked on this project because I wanted to dip my toes in the political science realm. I am an international affairs major and a more humanities focused one at that. And so I wanted to, try something new, do something a little bit more qualitative. And while I did enjoy this project, I think it did teach me a little bit more about my interests. And I think I do want to focus more on cultural studies. And so I think my next research will probably be centered more on the Francophone world because that's something that I'm more interested in. And so I think I would want to go to grad school for some form of cultural studies in the future.
Great. And then our last question, were you the original group working on this, and how does it connect to continuing research within this project? I think you kind of talked a little bit in your previous answer.
And so for this project in particular, me and Devin George were the first students to start coding for cases. Actually, Mr. Bolt had just written the code book for us at this time, but he still has many more cases that he wanted to code, even after I left the project. And so I'm sure that he would still be looking to hire more assistant researchers. How does this connect to continuing research within this project? So Mr. Bolt is doing a pretty wide-ranging project on human rights abuse denials. And so I think one of his more recent contributions to his overarching project was a study on Filipino public opinion on human rights denials within the Philippines. And so he did a survey experiment there, and he was able to include that in his overarching project. Like I said, this project is meant to fill some qualitative gaps that we have within this area of human rights. And that is what Mr. Bolt is trying to achieve. And so I think overall we did a pretty good job in adding some numbers to the field.
Awesome. Thank you, Catalina. And thank
Thank
you for presenting your research with us.
Of course.
Our next presenter is Emily. Emily, whenever you're ready, feel free to share your screen.
Sounds good. Hello, everybody. My name is Emily, and I am presenting my honors thesis research today. I worked alongside Dr. Kelly Klein for a little over a year now, close to a year and a half on this project. And she is in the realm of social stereotyping and social biases, and that's how I got into this realm of research. And so for our research topic, we wanted to focus on using something called the stereotype content model. This is also known as the SCM, and it assesses social stereotyping for working parents and how gender bias and ageism can affect their workplace outcomes. So workplace outcomes is going to be things such as how easily you can be hired, how easily you can be promoted, getting bonuses, financial incentives, and things of that nature. And so when we look at the SCM, it's visualized into this model that has 4 different quadrants, and it categorizes stereotyping into warmth and competence dimensions. And so warmth is going to be things such as how nice you are, how friendly you are, how approachable. It's also associated with some caregiving abilities as well, how well you handle children. And then competence is going to be things such as intelligence, capability, usually associated with career, success, and things of that nature. And this can be influenced by social factors such as age, gender, religion, race, and social class. And it identifies 4 different combinations, each with trade-offs affecting social perceptions and outcomes. So for example, if you look over here in the top left corner, this is the combination of high warmth and low competence. And it evokes feelings of pity because they're pitied for being viewed as incapable or incompetent, but they would be someone that you'd want to approach and be friends with because they're high in warmth. Whereas if you look at the bottom right, that's a combination of high competence and low warmth, and they provoke feelings of envy due to their high intelligence that's envied, but not how their demeanor is. So their demeanor is viewed as cold. And so the SCM highlights the complexity of stereotyping, emphasizing warmth's association with cooperativeness and competence with social status. And so when we look at the previous literature with working parents, taking parental leave can affect perceptions of warmth and competence differently for mothers and fathers, contributing to the motherhood penalty that can hinder women's career progression compared to men. The motherhood penalty basically is working women who start off in the career field, then they go on leave to have children, they come back and they get penalized for having children because it's assumed that their main job is going to be in their caregiving abilities. Hence, they're limited in being offered promotions, being offered bonuses and things of that nature. Whereas for men, they gain when they come back from having children because it's automatically assumed that they're going to be the main breadwinner. Hence, they're usually offered more promotion and more financial incentives. And so achieving job equality requires structural changes, such as providing temporal flexibility for working mothers, challenging biases favoring fathers, and addressing societal pressures associated with traditional gender roles. So temporal flexibility is basically just allowing more independence of working moms to adjust their schedule as needed so that they can balance both home life and career life in a better way so that they can also be deemed as eligible for promotions and things of that nature. And so moving on to ageism, discrimination against older individuals is pervasive due to negative stereotypes about aging, impacting job opportunities and leading to over-accommodating behaviors and benevolent ageism. This is basically baby talking older adults or automatically viewing them as incapable just due to the fact of their age alone and viewing that they can't do a job well. And then older individuals, particularly women, face gendered ageism, challenges including societal pressures regarding appearance and career advancement, highlighting the need for inclusive efforts to combat age-related discrimination. So gendered ageism is basically just the social factors that women face compared to men, and then that's intertwined with being an older individual compared to being younger. And so we move on to our final one of female professionals. Female professionals experience discrimination based on their physical appearance. This is known as lookism, and this intersects with previously mentioned gendered ageism. And this bias affects their recognition and authority as they may be perceived as either too old or too young, regardless of their actual age. And so when me and Dr. Klein looked at the previous research for this, specifically with female entrepreneurs, what we see is that that field is heavily critical of women rather than with men in that field. So it was constantly not being able or not being viewed as good enough. So there was always something to complain about of too tall, too short, being too masculine, being too feminine, wearing too much makeup, wearing too little, not wearing heels, like it just nothing was good enough. And so women in leadership roles encounter biases rooted in societal norms and expectations, leading to lower evaluations compared to men. This bias persists even as women gain experience, highlighting the ongoing need to combat discrimination and promote gender equality in leadership positions. We specifically see this on a global scale when we look at CEOs and how only about 5% of CEOs across the globe are female compared to male. And so when we look at my variables, we had gender and we had age. And so we had 2 male vignettes that were used in conjunction with age. We had 2 female vignettes that were used in conjunction with age. And then we had 2 younger vignettes used in conjunction with gender. And then we had 2 older vignettes that were used in conjunction with gender. And so what this means is that we had an older male, a younger female, and an older female. And so these are what those 4 conditions look like. So condition 1 was Dan at 28 years old with 7 years of work experience. We had condition 2 with Dan being 56 with 30 years of work experience. We had condition 3 with Kate being 28 with 7 years of work experience. And we had condition number 4 with Kate being 56 with 30 years of work experience. And so when we look at our hypotheses, we first hypothesized that the younger vignettes would be perceived as higher in competence and lower in warmth. For hypothesis number 2, we hypothesized that the older vignettes would be perceived as higher in warmth and lower in competence. And then hypothesis number 3, that the male vignettes would be perceived as higher in warmth and competence than their female counterparts, regardless of their age. And this was concluded of how we got these hypotheses were based off of that previous literature that I mentioned. And so for our study design, we had 134 participants, and we had a 2x2 between subject study design. So we had 2 levels of 2 variables. So we had gender, which was male and female, and then we had age, which was younger versus older. And so for our between subject study design, we basically took those 4 conditions that we had, and we randomly assigned one of those 4 to each of our participants. And we had 7 questions that assessed warmth, and then 15 questions that assessed competence in those financial outcomes and hiring outcomes. So we had 3 parts to our survey. The first part was we had Likert scale questions that were used to rate the candidates on SCM traits. Then after that, they completed a demographics form that they filled out. And then part 3 was additional questions that were given to participants who had children. And so when we look at our warmth data first, what we found was that when we looked at the trustworthy question, we had a main effect for gender and not gender, but for age, where our older targets were rated to be more trustworthy than our younger targets. And then when we move on to main caregiver, this was the strongest statistical correlation that we had above all of the other ones. So basically we asked them, how likely would you be to assume that this person is the main caregiver of their family? And what we found was a huge main effect for gender. And so our female targets were rated to be far more likely to be those main caregivers than the males. We had a p-value of less than 0.001, which is fairly significant. And then when we move on to overall warmth, which basically means we took our 7 warmth questions, we took that data and into a statistical software, we overlapped all of them, and then it pumped out with an overall warmth number or output. And so we also had a pretty strong correlation for that as well, with a main effect for gender. So our female targets, again, were viewed to be more warm overall than their male targets. And so when we look at our graphs, when we visualize this, we look at main caregiver. So all of the gray bars are going to be the female targets, and all the black bars are going to be our male targets. And so when we look at that stark distance between the two, that's basically saying that main effect for gender of the women are viewed to be the main caregiver compared to the males. And then when we look at average trustworthiness, the reason why those 2 bars move up is because of that main effect for age, where those older targets are viewed to be more trustworthy. Then when we move on to competence, for skillfulness, we had a pretty strong correlation as well. So we had a main effect for gender, where our female targets were viewed to be more skillful than our male targets. And then when we move on to our leadership position, how likely they would be considered for that, we had a main effect for age. So our older targets were viewed to be more likely to get that position than the younger ones. And then for hiring the consultant, we had a main effect for age, and we had a main effect for gender. So our older and female participants were rated to be more likely to be hired than our younger and male participants or targets. And so when we look at that of hiring the consultant, again, we see that stark distance between the 2 bars, which is showing that gender. And then when that black bar meets up to that gray bar, that's again showing that age main effect. And then when we move over to average skillfulness, that distance between those 2 bars is again showing that main effect for gender. And so our conclusions state that conclusion 1, our older candidates had higher ratings for trustworthiness than their younger counterparts. Conclusion 2, our female candidates were perceived as more likely to assume the primary caregiver role within their families. Conclusion 3, our female candidates were perceived as more skillful than their male counterparts. Conclusion 4, our older candidates were perceived to be more suitable for leadership roles than the younger candidates. And conclusion 5, our older and female candidates were more likely to be hired when compared to the younger and male candidates. And so for our limitations, we had a small sample size. If we were to replicate it, we'd love to have more participants to take it to further analyze our results. The next limitation is we had a sample distribution that was disproportionate. So majority of our participants, around 70-ish%, were women compared to men. We'd like to have that be more proportionate. Third was our age demographic. So this was more of a learning curve for me in terms of research of you never group a continuous variable. And so if we were to replicate it, of course, we'd keep age of our participants as a continuous variable. And then for our further limitations for outreach, again, it was mostly within FSU, so a southeastern region. And this could have influenced our potential findings due to regional biases as well. If given more resources, we would love to have it be more nationwide so that we could get a better grasp of the holistic view of how the US is viewing fake vignette candidates of people. And then for future directions, future research should explore social factors that could impact perceptions, such as multiple marital status conditions or racially ambiguous names. Replicating and varying this study design is crucial to show significant social biases and to integrate these findings into managerial and hiring training programs to foster self-awareness and mitigate bias effectively. And then this was my committee for my honors thesis. I'm just thanking them again for all that they've done for me, and I will be taking questions.
Were any of the conclusions surprising to you or your team?
Oh, 100%. We were expecting it to be the males who were getting all of the positive attributes when it came to hiring or any of those benefits. And we actually got some pretty alternative results where we kind of got the opposite effect besides our main caregiver role and that overall warmth, which we expected. But overall with hiring and stuff, women overall came out on top. However, there are a lot of limitations to this study and the fact of lack of resources. Hence, we can't fully confirm or deny what we got just because of our sample base was mainly college women, and they could have had an in-group bias towards our targets and our female targets specifically.
And do you think doing this research impacted how you view age or gender within a work setting now versus beforehand?
Ooh, that's a good question. Yes and no. On one hand, before going into the research, I definitely felt like there definitely was a gender double standard along with an age double standard. But I didn't know it was this bad. So when I did the full literary analysis and everything, it was quite sad to see what the previous research has showed with that, and especially now. So I think it just reconfirmed that, but also I definitely didn't expect it to be so sad, I guess. But we were very happy to see more progressive results with this. So hopefully things are turning in a more positive direction for that, of stereotypes regarding that aren't as stark anymore. Hence, women and older adults won't have as high barriers to entry when it comes to the workplace, especially now in the economy where older individuals are retiring at much older ages or not retiring at all. So destigmatizing those stereotypes is very crucial for our workforce and how we treat people in the workplace.
Great, thank you. Any more questions? Here we have Latika, very interesting. I might have missed this, but when was the SCM model first developed? It seems very useful.
The SCM model was developed by Cuddy and Fisk. Cuddy basically was a student working under Fisk. They're very well known. I think they're Harvard, or they went to Harvard or somewhere around there. But it's a very well-known thing in social psychology. I think it was developed in I think it was around like the 80s, 90s, somewhere around there. And it's drastically developed and changed over time. So what we did was we basically took that model, and the vignettes and the questions that we asked were taken or used from Cuddy and Fisk's studies and what they did and how they assessed warmth and competence. And then we took that and redirected the variables. So we did different variables to assess what we were aiming to do. And so that's basically what we did, is we basically revised their model and added to it.
Great, thank you. Okay, Emily, thank you so much for sharing. We really learned a lot from your research, and Emily's actually our last presenter for the day. So I would like to thank everyone for attending. Thank you to our student presenters, and thank you to our audience.